Hey everyone, this is Elad from Astrolab Diagnostics, and this is the first of a series of videos about immune monitoring, with a special emphasis on immune monitoring in the context of flow and mass cytometry. And in this initial presentation, I'm going to talk a bit about the background behind immune monitoring. Why do we need it? What is it good for? And some really essential basics on how to do it. Before I begin, full disclosure. First of all, I am neither a biologist nor an immunologist. I'm a bioinformatician in my background, so I am really not the expert on these kinds of topics. I'm doing my best to teach them and convey them. With that said, I apologize in advance for any inaccuracies. Please reach out to me if you have any suggestions on how to amend any of these slides. Next, I couldn't really find any clear definition of immune monitoring, and it seems to be one of these terms that when you talk to people, they have a very clear idea of what it means, but that clear idea might differ between institutions, researchers, and papers. So I'm showing just another opinion, and there's always this joke that there is no consensus in immunology, but this is as close as I could find myself in this context. So really brief background about the immune system, and this is a very high level. So on the most basic level, the immune system is built to protect the body against infection. The immune system evolved to protect the body against infection, and in this case, infection is any invasion by a disease-causing agent, also referred to as a pathogen. So this includes viruses and bacteria and parasites and so on, really any of these things. And since there is such a wide range of threats in this context, the immune system is composed of a staggering variety of cells and cell types, and I think I can safely say that it is the one system in our body that has the most varied and the most heterogeneous cell populations. Very briefly, there's 4 major compartments in the immune system: myeloids, NK cells, T cells, and B cells, and there are many sub-classification and sub-subclassification within them. Now, generally speaking, and you'll notice that I will be using this word a lot, generally, because there's always exceptions. But generally speaking, there's 2 major types of immune responses. The first one is innate immunity, which is non-specific and it's very fast. And basically, these are different cells, especially monocytes and NK cells, that can identify a wide variety of non-self agents in our bodies, that is, cells that do not originate from our body, and destroy them. The innate immunity is one of these things that works so well that you don't even know it's there, because we are constantly under assault by different pathogens, and I don't have the numbers, but I believe that the innate immunity takes care of many of them. Then the second one is called adaptive immunity, and the adaptive immunity is pathogen specific and it is slow. And briefly, the adaptive immunity evolves constantly to identify novel threats and customize elements of the immune system to attack them. And just very broad idea for one of the mechanisms involved is we have certain cells in our bodies that continuously generate homing beacons that can stick to specific pathogens, whether it's bacteria or virus infected cells. And once a cell is bound by this beacon, other cells in the immune system are going to go and destroy it. And these beacons can be customized through genetic engineering, basically. And it sounds very fancy because in many ways it is a fascinating process. However, for the purpose of this discussion, 2 types of immunity, innate and adaptive. Now, it is really important to point out that the immune system responds to non-pathogen events as well. So it was evolved to defend against infection, but there's evidence that the immune system responds to a wide variety of events. Pregnancy is a good example. We can see differences in the immune system of pregnant women, but even if you bumped your head or you had some physical trauma, you can sometimes see responses in the immune system. So in many ways, it responds to almost everything that our body encounters. And finally, the immune system can go wrong. And just a few examples: autoimmune disease are when the immune system doesn't discriminate between self and non-self and attacks our bodies. Allergic response is that when the immune system identifies an outside agent and responds too aggressively to it. And finally, a bit more artificial, but transplant rejection is when we like if a body in a patient's an organ in a patient's body fails and we would like to transplant a different one, and the immune system identifies it as a non-self and attacks it. So this was really immune system 101, and thank you for bearing with me. So what is immune monitoring? And I'm just going to read a slide here. Immune monitoring, and again this is my interpretation, is examining and quantifying the frequencies and states of the various cell subsets in the immune system and the interactions between them across milestones of interest in a patient's disease progression. So let's go over this definition and break it down a bit. The first part is that we'd like to examine and quantify. In other words, we are looking for trends that we can build hypotheses around. We would like to be able to say, in a given disease, here is what happens, and we can provide a very clear definition of what that thing is. The second part is the frequencies and the states of the various cell subsets. And as I mentioned, the immune system has a staggering variety of different cell subsets, different cell populations, and they have different functions and different roles in the immune system. And immune monitoring measures, first of all, how many of them we have. For example, in HIV patients, the amount of CD4 T cells eventually plummets, and also how they behave. For example, in certain disease states, such as cancer, CD8 T cells are going to be exhausted around the tumor, which means that they do not function. Then we would like to measure the interactions between them, because these cells communicate with each other through different mechanisms. For example, there's messenger molecules called cytokines that immune cells use to communicate, and it's possible that these will be somehow correlated with disease progression across milestones of interest in a patient's disease progression. And to me, this is the most important part. At the end of the day, we would like to help patients. We would like to make human health improve. And the focus, the emphasis should always be about a certain disease, a certain condition. And again, there's a huge variety here. It could be cancer, it could be autoimmune disease, it could be Alzheimer's or Parkinson's, it could be infectious disease. But at the end of the day, we would like to help people. And there are certain milestones, time points, stages across these infections that we would like to examine. So, why do we do this? Other than the sheer scientific joy of learning more about human health, what is the motivation and how do we tie this into a clinical outcome? So why do we do this? To reduce the complexity of the immune system into one or more biomarkers that, by means of regular measurements, provide insight into the health of the patient and contribute to some clinical benefit. And I highlighted the critical terms here, in my opinion. The first one is biomarkers, and biomarker is, much like immune monitoring, a very broad term. A biomarker is anything that we can measure and has some biological outcome. And immune monitoring, by its definition, is very broad. Going back to the definition, we're looking at different cell subsets, their frequencies, their states, their interactions. It's a lot. We would like to reduce this into a small number of biomarkers that we can measure easily. In other words, that a physician, your PCP or your oncologist, would be able to measure in a relatively straightforward way and use that information to make some sort of clinical decision, a diagnostic, anything that can improve the patient's life. So again, what do? We look at the cell subsets and in the immune system, their frequencies, their states, their interactions, and we're examining this wide variety in order to isolate a small number of biomarkers, ideally one, but sometimes more, that we can measure in the clinic. And let's talk about some examples. So one common example is in immunotherapy. And immunotherapy is a very exciting treatment direction for cancer. The idea is that we can teach the immune system to attack the tumor. And over the past few years, we noticed that the scientific community has noticed that this is extremely effective, usually. When it is effective, it is extremely effective. And there's now this separation into two types of patients, responders and non-responders, where for responders, immunotherapy is extremely efficient, while for non-responders, it practically doesn't do much or even anything at all. So one potential application for immune monitoring is within the context of the clinical trial of immunotherapy to continuously monitor the immune system of the patients in a prospective way. And then when the trial is done, we can know which of them responded well and which of them did not. And we can use this to build a model that can predict whether this type of immunotherapy will work for a given patient. And now for future patients, instead of taking all of them and training them with immunotherapy, we can apply this model, take these patients that we predict are going to be responders, and just treat them. And like any kind of treatment, immunotherapy costs money. Immunotherapy causes agony and anguish to patients, and we would like to make it more targeted. And here's another example, which is much more recent. In fact, this paper was published a few days ago, and the world is currently dealing with a worldwide epidemic of COVID-19, which is caused by the SARS-CoV-2 virus. And there's this paper from Lu and Jiang that shows, it's a team in China and New York, and they show that the virus can infect T lymphocytes and can destroy them. However, this study, as far as I know, was done in a petri dish. With immune monitoring, we could take COVID-19 patients and assess their immune system continuously throughout the infection to see whether there is a decrease in the population of T cells. And the ramifications for this are extremely wide as far as treatment of the disease and its symptoms are concerned. Now, the last question is how to do immune monitoring, and I am going to elaborate on this in future talks, and specifically I'm going to focus on one branch of technologies called flow and mass cytometry. And before I talk about the how, I would like to draw on a comment that was done in this paper by Sood and Testro, specifically in the context of immune monitoring of liver transplantation. And what they're saying is that we need a variety of tests or assays, and we need a clinician to be involved. So on a very broad level, how to do immune monitoring? Well, take multiple different technologies, use them to assay different parameters, different potential biomarkers. In other words, test a large number of hypotheses, and then bring in the clinical knowledge to decide which of these is the most relevant. And here is one favorite example of mine, and if you've listened to any of my videos, you've seen this slide multiple times. And this is an immune monitoring trial that was done by Professor Adib Rahman and others as part of the Human Immune Project Consortium. And in this study, 42 pediatric patients who were sick with the chikungunya virus showed up at the clinic in Nicaragua, and blood samples were taken from them during 2 stages of the infection, the acute stage and the convalescent stage. So we have these 42 patients, and we have 2 different time points, and we would like to compare the structure of the immune system. We would like to draw biomarkers that allow us to identify, for example, when the disease has subsided, or what kind of responses the disease elicits within the immune system. And this study has several different assays or technologies involved. There's RNA sequencing, there's a 40-plex cytokine assay called Luminex, there's a titer of CHIKV, which is the marker of the virus, and there is mass cytometry with a 33 marker panel. So there's 4 different technologies involved here and different clinical covariates that were combined in order to assess and identify biomarkers for the disease. In future videos, I'm going to focus on just one of these technologies, namely mass cytometry and its older sister, flow cytometry. So please join me in these future videos. I will post them on my YouTube channel in the coming days. Please subscribe to the YouTube channel and please subscribe to us on LinkedIn. All of the links are going to be in the video description. This is a great opportunity to learn about immune monitoring, which is already revolutionizing the way we do medicine. Thank you for your time and have a wonderful day.